Good morning, good afternoon, and good evening, dear listeners. Professor Dr. SKA is back for you with his podcast, Immunology for Everyone, episode number 5. For all those times we've had fever, for all the times we have had those inflammatory symptoms of running nose, cough, well, we should be thanking our body for letting us know that it is fighting some foreign invasion. With the body's immune system hard at work, it is next our work to get all the right investigations done and tackle the pathogen appropriately. Let us understand more today as to what is happening and at what stage. Your questions keep driving interest and are most welcome anytime. Don't forget to like and share these podcasts with your friends and peers.
Welcome back to this podcast. So, guys, whenever we have any infection, the infection leaves certain kind of immunity in our body. We have seen that sometimes the immunity lasts for lifelong because it doesn't allow the same pathogen to infect you again in your life. Like take the example of smallpox, chickenpox, measles. So they induce a lifelong immunity. Whereas other viruses or pathogens, they induce a short-lived immunity, like we take the example of SARS-CoV-2. And we have heard that there were many repeat infections and breakthrough infections after the person has already recovered. So, but one thing is sure, that this immunity is antigen specific or pathogen specific, and it carries some kind of memory. So what is the kind of immunity which does that, and what are the features of this immunity? And that is what we are going to talk about today, the adaptive immune response, which is the antigen specific immune response. So what are those cells which drive this immunity? There are some specific cells in our body which are called the T cells and the B cells. They are the cells which brings in the antigen specificity. Now, for this purpose, now we know that any pathogen or a molecule which is able to induce an immune response in our body, we can call it an antigen. So, for all practical purposes, we'll be calling it as antigen because it will induce the immune response. So, talking about the T cells and the B cells, where do they come from? They are the lymphocytes. They are present in our blood in abundance and they are circulating. They are a part of our white blood cells. These cells, all blood cells, they develop from a common progenitor cell in the bone marrow, which is the hematopoietic stem cell, which is the pluripotent stem cell. Some of these cells, they develop into T cells because they go into thymus for further development. And all those cells which develop and mature in thymus, they are called the T cells because of the reason that they are developing in thymus. So there are many factors, and thymic education leads to the development of these cells into mature T cells. So all these T cells, they express a specific receptor on their surface, which is called the T cell receptor. But these T cell receptors are so diverse that each T cell will express a T cell receptor with this different antigenic specificity because they have to handle so many infections throughout their life. So each T cell receptor has a different specificity for a different molecule. So they recognize a different epitope, which we call the T cell epitope. Okay, and all these T cells, they also express a CD marker, which is CD3. Okay, so during this education, they also further mature into different lineages like CD4 cells, which are the T helper cells, T suppressor cytotoxic cells, they are the CD8 cells, and so on and so forth. On the other hand, the B cells, they develop in bone marrow. They also express antigen specific receptor, which are called the B cell receptors, and they also have similar diversity, and each B cell expresses a immunoglobulin molecule which has a binding capacity to a different molecule. So they also have antigen specificity. But the difference between a T cell and the B cell is that T cell receptors, they cannot recognize a molecule or an antigen de novo. They always want that the antigen has to be presented to them in association with a MHC molecule. We already talked about MHC molecules, the MHC class 1 and 2 molecules. They are very important. And here comes the importance of these MHC molecules. So any peptide which is loaded onto the MHC class 1 or 2, they are the only antigens which can be seen by the T cell receptor. And these peptides are called T cell epitopes. They are normally short peptides from 8 to 15 amino acid short, and T cell receptor basically recognizes two things. It can recognize a foreign molecule, which is the T cell epitope, along with a self MHC molecule. So a cell which is expressing MHC class 1 or 2 molecule along with the T cell epitope, they can activate the T cells. So on the other hand, B cell receptors, which are the immunoglobulin molecules, they can recognize soluble molecules, soluble peptides also, or the globular proteins also. So these molecules they don't require any presentation through MHC class 1 and 2. So how do the T cells get activated? What are those cells which perform that function? They are the cells of phagocytic nature. We all have discussed that there are phagocytic cells. They are present in the periphery, and they are able to engulf the pathogens or the invading microorganisms, and they have the capacity to degrade them by all their effector mechanisms. And then inside these cells, they also express MHC molecules, where the pathogen molecules are loaded onto the MHC molecules, class 1 or 2, and they are expressed on their surface. So these phagocytic cells, they perform the function of antigen presenting cells. And the professional antigen presenting cells include the macrophages and the dendritic cells. The dendritic cells basically are more potent antigen presenting cells because they have the capacity to activate the naive T cells. Now, there is one thing more which is very important, that all those pathogen associated molecules or pathogens which are acquired exogenously, like bacteria by phagocytosis or endocytosis, then the antigens of these pathogens, they are loaded onto MHC class 2, and they are expressed along with MHC class 2, and they will recognize CD4. They will interact with CD4 positive cells, because CD4 molecule acts as a co-receptor for MHC class 2, because there is a binding site for CD4 molecule on MHC class 2. Whereas all the molecules which are expressed endogenously, like tumor antigens, viral antigens, they get loaded onto MHC class 1, and they are expressed outside. And all these molecules which are expressed along with MHC class 1, they will go and activate CD8 positive T cells, because CD8 molecule has a binding site on MHC class 1, and so CD8 molecule acts as a co-receptor for them. So we have this antigen presenting cell, which has all the characteristics of being a antigen presenting cell, because it has the capacity to engulf the antigen, degrade it, then the molecules are loaded onto the MHC class 1 or 2, they are expressed on the surface, and they can present it. And then there is another most important characteristic of these cells is that once they look at the pathogen associated molecular patterns through their pattern recognition receptors like TLR and other things, they start expressing many molecules on their surface, which are called the co-stimulatory molecules. They are like CD80, CD86, CD40, and many others. So these co-stimulatory molecules are very important for passing the activation signal to the T cells. So these antigen presenting cells, they also express these. So dendritic cells, which are initially called the immature dendritic cells when they are present in the periphery, once they acquire the antigen, they process it and they express the antigen on their surface. They also express, they start migrating from periphery to the lymph node, and where they wait for the T cells to come and present the antigen. And the T cells in the thymus, once they are mature enough to ready to look at the antigen, they also migrate from from the thymus to the lymph nodes, and here they come and start scanning all the dendritic cells which are expressing a foreign molecule along with the MHC molecule, and if they don't find these specific molecule expressed, the T cells will go away into circulation again and come back again, and till a T cell with a receptor which finds a specific T cell epitope being presented by the MHC molecule, then the T cell and the B cell, they interact with each other and get bound to each other. And this TCR, T cell receptor interaction with the MHC molecule carrying the foreign molecule is called the signal 1. But this signal is not sufficient to activate the T cell. The T cell absolutely requires a second signal for activation, which comes with the interaction of a co-stimulatory molecule with its counterpart, which is expressed on the T cell. So T cell express many molecules, and the antigen presenting cell also expresses many other molecules. So the T cell and the antigen presenting cell, they come together, and that leads to the formation of immunological synapse. And this immunological synapse hold the two cells together for quite some time, and then the T cell gets the activation signal, and once it gets the activation signal, it starts proliferating. So there is a clonal expansion. So this particular T cell carrying a specific T cell receptor clonally expand, and then it differentiate into different lineage of cells like Th1 type, Th2 type, Th17 type, T regulatory cell, depending on the cytokine milieu. Because antigen presenting cell, while presenting the antigen, also releases many cytokines and creates a cytokine milieu in that microenvironment. And the T cell, which has seen the antigen now, will differentiate into a specific kind of cell depending on the cytokine milieu. If the cytokine milieu is like IL-12 and IL-18 released by these antigen presenting cell, then the T cell develop into a Th1 type of cell. And similarly, if it is IL-4, it develops into a Th2 type. So similarly, this differentiation occur, and then all these T cells, once they get activated, it will go and release many cytokines, and then these cytokines will go and activate many other cells, including B cells. So there is a T cell B cell interaction, and that leads to the activation of antigen specific B cell, which go on to differentiate into plasma cells, which make the antibody, which has got the specificity for the same antigen which has been presented by the antigen presenting cell. So this is a very interesting story about T cell B cell crosstalk or interaction, which leads to generation of specific antibodies. We'll talk about that in the next episode. For the time being, we should understand few things. That there are antigen presenting cells, which are basically professional antigen presenting cell. We have the macrophages, we have the dendritic cell being the most potent antigen presenting cell. B cell also having the surface immunoglobulin, they can bind to the you know, uh, B cell epitopes or globular proteins, they can also bind to the pathogen because they have the surface immunoglobulin, and as they bind it, they can endocytose those pathogens, and they have the degrading machinery also. They also express MHC class 1 and 2. So B cells also can be a good antigen presenting cell. So at times they also behave as antigen presenting cell. So we have three kinds of antigen presenting cell: the macrophages, the dendritic cell, and the B cells. Whereas some other cells in the body which have got these characteristics of antigen processing and presentation along with MHC class 2 molecules and co-stimulatory molecules, they also act as antigen, but under various circumstances. So most potent are the dendritic cells. So for the time being, we should understand that the T cells have got activation signal, and that requires two signals. One is through TCR and MHC molecule. The second comes from the co-stimulatory molecules. And there is a third signal which comes from the cytokines. And all these signals, they lead to the activation of T cells and its differentiation. So we'll close here. And next time we'll discuss about how T cells and B cells interact and leading to the formation of antibody forming cells, and also how the memory is generated, how some of the cells become memory cells, which are long-lived cells. So till then, thank you very much, and I'll be happy to take up any questions if there are.
Wow, this is an amazing insight into all this activation phenomena going on, and immunology has never ever sounded so interesting. Thank you, SKA. We have just the time for one question, which has come through our email. The T cells develop in the thymus, and the antigen presenting cells acquire antigen in the periphery, and they both migrate to the lymph node for interaction and for antigen presentation. How do these two cells at two different locations get to know where to go?
Oh, thank you very much for this question. Very nice. I think this is because the T cells in the thymus, when they are maturing to as naive T cells, which have to see the antigen, they also start expressing a chemokine receptor, which is called CCR7. So the chemokine for CCR7 is CCL19 and CCL21, which is expressed in the stroma of lymph nodes. So these cells, the T cells in thymus, which are expressing chemokine receptor CCR7, they get chemo-attracted to lymph nodes. So they migrate from thymus to lymph node and home there. And similarly, the antigen-presenting cell, once it acquires the antigen and start expressing co-stimulatory molecules and MHC molecules, it also start expressing CCR7, which is the chemokine receptor, and it also gets chemo-attracted, and it also goes and homes into the lymph node. So both the cells at different location, but they express similar chemokine receptors, and so they move and home into a common place, which is the lymph node, and that brings them together, and the lymph node becomes the site of interaction between antigen presenting cell as well as the T cell.
Thank you very much. Sign off for today. See you next time.